and a warm welcome here from our headquarters in London. Good evening to our viewers in Asia and Australia. Good afternoon to viewers in Europe and Africa, and good morning to the Americas. I'm Torben, Chief Business Development Officer for Lifebit, and I'm delighted to be your host for today's webinar. And the reason for this webinar series? Well, Lifebit is a global leader in genomics and health data software, and we're empowering organizations across the globe to transform how they securely and safely leverage sensitive biomedical data sets. We are committed to solving the most challenging problems in precision medicine, genomics, and healthcare. And our mission is to create a world where access to diverse biomedical data will never again be an obstacle to curing diseases. So for these webinars, we're bringing together pioneers and experts in genomics, biotechnology, and population health to inspire, to challenge, and hopefully to delight. So a little bit of housekeeping for today. Please do post your questions in the Q&A tool here in the Zoom chat. Sorry, not in the chat, put it in the Q&A tool, and we will try to get to as many questions as possible towards the end of the talk. However, can't promise that we will get to all, because we're delighted to welcome more than 120 people in today's webinar. And very fitting for today's topic, these are diverse also in background. We have attendees from over 40 organizations, stretching sectors such as government, academia, biobanks, pharma companies, and policy groups. And even more exciting, these attendees are representing over 30 countries across the world. It's a true reflection of the global nature of this field, and also Lifebit work that was already spanning 5 different continents. And from around the world, there are also a fantastic set of speakers, and we're looking forward to a really engaging discussion and also engaging with your questions. All 4 of today's speakers are experts in their field and represent organizations across the world with the mission to improve the health and lives of patients, as well as the overall populations of their countries. And the first speaker I would like to introduce is Professor Lygia da Veiga Pereira, the CEO of GenT. Lygia founded GenT in 2021, a biotech company that is leveraging the genomic diversity of the Brazilian population to foster research and innovation in the life sciences industry. She has also a full professorship in genetics and evolutionary biology at the University of Sao Paulo. And she has a PhD from Mount Sinai Graduate School in New York and has established an impressive career in human genetics research, ranging from stem cell research to the DNA do Brasil project, a population genomics initiatives she launched in 2019. I personally had the distinct pleasure of knowing Lygia for over 4 years when we first discussed collaborative platforms in genomics at the OECD conference. But enough from me, and over to you, Lygia.
So Torben, thank you so much for the opportunity to be here, and I congratulate Lifebit on this initiative to bring awareness of the importance of data diversity in genomics. I'm going to talk about how we're leveraging really Brazilian diversity to accelerate innovation, and why talk about diversity. So probably everybody who's here watching knows this figure, and there is this large and growing disparity between the proportion of European and non-European genomes being sequenced and analyzed, and this Eurocentric bias leads to various problems, especially regarding the validity of GWAS studies to non-European populations. And so the implementation of precision medicine in non-European populations. And moreover, these understudied populations they're like the low-hanging fruit for genetic discovery, because they will contain variants common in these groups, and there are very rare or absent in European populations. So today, I will tell you about the value of the Brazilian population to increase diversity in genomic studies. We have a very large population, 500 years of admixture of Native American or indigenous populations, African and European populations. And I will talk with more detail about this right now. So a bit of our history. Before pre-contact with Europeans, there were 5 million people in Brazil and divided in several distinct linguistic groups. And then the Portuguese arrived. And in the years 500, 1500, they forcibly brought millions of enslaved people from the West and East Africa. And there were also other large migrations from mostly Portuguese, but other European populations. And in these last 5 centuries, these deeply diverged human continental populations have come together in the America, and leading to this unprecedented scale and rate of admixture in human history between these previously isolated continental populations. And this was very elegantly shown by the Candela study, this consortium of the analysis of the diversity and evolution of Latin America. They genotyped almost 10,000 individuals from Latin America, and 1,700 of those were Brazilians. And here we can see, and we look here at the populations. There are based populations, and we can see the different contributions of Native American ancestries, genomic ancestries, and different European ancestries. And we see how Brazil has a very different European fractions than the rest of Latin America because of the Portuguese, the division of the continent between Portugal and Spain. And we can also see in the south, we have the contribution from other European populations. And finally, we have this large contribution of African populations, both West and East African throughout Brazil. Brazil also received in the 1950s a large immigration of Japanese, and today we have the largest Japanese population outside Japan. So in 2019, we launched the first whole genome sequence-based population genomic project in Brazil called the DNA do Brasil, DNA of Brazil, which is now led by my colleague Tabitha Hunemeyer. So our strategy at the time, due to limited funding, was to jumpstart this project, and instead of constructing a new cohort that would have to be clinically characterized, we took advantage of several smaller cohorts of Brazilians that had already been clinically characterized. We had some that were disease-specific, others were populational, but the important thing is that it contains people from all different regions of Brazil. And I'm going to show you now some of the preliminary data that we generated. And this preliminary analysis demonstrates that Brazilians exhibit this continuum of genetic variation between the continental source populations, African, Europeans, and indigenous. And the highest average contribution comes from European ancestries, but you can note that the amount of Native American ancestries in this sample is almost 13%, which is considerably higher than the estimated for the Brazilian population in the past, which was from 7 to 9%. So this shows that it's just a matter of going to different regions, and we can find these increased proportions of indigenous ancestries. And the other interesting thing, and this is what we show here, these ancestries according to the different cohort studies. and how they vary among studies because of the respective geographic location. So this is a population study from the north region of Brazil, the Amazon region, and then we can see higher proportions of indigenous genomic ancestries. While others in the south, for example, this one in the south of Brazil, we have an increased proportion of European genomic ancestries. So finally, we compared the variants in the DNA do Brazil data set with other public whole genome sequence data sets, and it reveals a high number of novel variants, more than 11 million. And most of them are rare variants, exclusive of this Brazilian population, and that's mostly coming from the indigenous and the African ancestries that we captured. And so this shows that the Brazilian genomes have a great potential for new insights. And then these findings from the DNA do Brasil project gave us a sense of urgency to develop a novel platform with harmonized data. And remember, DNA do Brasil is this patchwork of different cohort studies. So we want to develop a novel platform with harmonized data from hundreds of thousands of Brazilians, both because of the need to include our population in precision medicine and because of the opportunity for innovation based in our diversity. And this is when we launched GenT. So GenT has this mission to include Brazilian population in precision medicine, but we also see our genetic diversity as an opportunity to advance biomedical research and innovation. So I apologize for this busy slide, but what we're doing is we are creating a new cohort of Brazilians. And so we make this, we recruit participants, making partnerships with different health systems, either private or public. And in these health systems, they help us spread the word about our program, and we do the recruitment and the onboarding. And that involves of course, the consent, a very extensive questionnaire about health and lifestyle, and then anthropometric measures and a whole bunch of blood exams. And this is, and also the samples for our biobank. And eventually we keep the participants engaged because we want to follow them longitudinally through a digital platform, and that they can access in their cell phones, where they will receive all the lab tests and health reports, and through which we can keep them engaged in the project and send them further questionnaires and add more phenotypes that we can capture. And all of this is building our infrastructure. So we started recruitment. We're in the early days. We started recruitment at the end of last year. We're reaching 5,000 participants now, and we started close to home. But we are moving now away from the southeast region towards the northeast and the Amazon region in order to increase the proportions of non-European genomic ancestries in our cohort. And so this platform will be used for countless, to answer an infinite of questions regarding history, biology, health, and we built it to maximize its value in terms of being able to recontact these individuals and store longitudinal plasma samples so that we can, in the future, this allows for different omica analysis of these samples. And so this is our long-term vision. So right now we're really building the infrastructure. We Our goal is to reach 10,000 individuals recruited by the end of this year, 25,000 by the end of next year, and within 4 years to reach 200,000 participants enrolled in this population-based recruitment. And so this is the infrastructure. And as time goes by, and as we build the platform, we are positioned to be this strategic partner for pharma and biotech who want to leverage this data for innovation. And with that, I finish, my presentation. And again, I thank Lifebit for the opportunity and to talk about the diversity in the Brazilian population.
This is hugely insightful, and it's such a phenomenal effort that you've been and your whole team have been building there. I want to jump to one question right away, which is kind of at the burning heart of all of this. You presented, of course, the great potential that data from Brazil has, but what does genomics research as a whole really has as the need for diversity? So why are we talking about diversity as a pure hard need? What makes genomics research more effective when it looks at more diverse data?
Well, we need diversity because we need to understand the biology of Homo sapiens, the world's population, and not just of a subset of the world population. So both in terms of equality and medical access to precision medicine, but there's also this issue of science. We can learn a lot about human biology, understanding the genomics of all the diversity that we have in the world. So if we don't do that, this is a missed opportunity.
Absolutely. I couldn't agree more. Again, thank you, Lygia. We'll be coming back to you as part of the panel, but now we are moving up from South America to North America, and it's a great pleasure for me to introduce Victor Angel, CEO and founder of Omica.bio. And Omica.bio is a community-driven biobank that is based in Mexico City, and is on a mission to bring the Latinx population to the forefront of precision medicine research. Previously, Victor founded Entheogen Genome Project. That's a non-for-profit genomic data consortium for the conservation of fungal species. And he's also served as the CEO of Mori Biosciences, a natural product discovery company. But for now, we're jumping straight to Victor. Over to you.
Thank you so much. Thank you everyone for being here. It's really it was a very pleasant surprise to hear about this webinar, given Lygia and everyone else in this sector, especially in Latin America, the need to really have a platform to speak about our work, especially in an international stage. So thank you very much for having me. A specific approach that we're taking at Omica to overcome the obstacles around genetic diversity. So first of all, our mission, as you well said, is to bring Hispanic Latino populations to the forefront of biomedical research and development. Now, before jumping into the diversity bit, a few days ago, I was doing a back-of-the-envelope calculation to try to figure out how much of the human population has been studied by genetic studies. And it's appalling to see that even in the most optimistic calculations, we're still at less than 1% of the global population in terms of representation in genetic data sets. So this means that everything we know about the human genome to date, every precision therapeutic that is being developed, every predictive algorithm, is really coming from a very minuscule sample in relation to the vastness of humanity. And this back-of-the-envelope calculation was basically done by analyzing the prospective studies that are going on around the world, namely, in US, Canada, Europe, Asia, the All of Us research program that's happening in the US, the Million Genomes project that's going on in China. And even when we consolidate this entirety of projects and data that is being generated in the global north, we still find that diverse populations, particularly Hispanic Latino populations, still account for 0.25% of total GWAS representation. So I imagine Matt is going to go into deeper details as to why this is the case, and Lygia did a really good presentation onto why this is relevant. But to us, the most present challenge is data biases, right? So the fact that there is a lack of representation in GWAS studies really leads to a data cascade, especially today in the age of AI, where we have really complex computational models being developed based on massive genomic data sets. AI models that are being used for AI drug discovery, models that are being used for clinical decision support. All of these models are based on data that is mostly white European data. So not only is this a problem in terms of accuracy, but it is a problem in terms of health disparities. The way that we see data models is as them having compound effects, right? So when you develop a model that is constantly learning based on the feedback that it's receiving from the outside world, you start to see that we're going to have models that are increasingly more efficient in white European populations and increasingly less efficient in more diverse regions of the world, namely Africa and Latin America. So how do we flip the narrative, and how do we start to see these regions not only as regions that have fallen behind in genetic research, but as regions that are untapped source of genetic insights? So if you take a look at this map representing the percentage of European admixture in the South American and North American population, we see that regions from Mexico, Southeast Mexico down to Colombia, Bolivia, Ecuador have an incredibly low percentile of European admixture, speaking to the vastness of potential genetic insights that are yet to be seen by the scientific community. So, for instance, Lygia's research in Brazil is clearly demonstrated as having, a considerable percentage of European admixture, while still offering over 11 million unique variants to the Brazilian population. So if we extrapolate this data to the rest of the region, we really see that Latin America is not only a place where we need more genetic research to improve upon health disparities, but really a region that may hold secrets to potential cures, treatments, algorithms, etc. So really the question is, how do we do it? How do we bring the Latino Hispanic population to the forefront of biomedical R&D? If you're from Europe, one of the suggestions that will probably come to mind is ask your institutions, right? And in Mexico, at least, and I think this is representative of other countries in Latin America, the budget for the Mexican Institute of Genomic Medicine was cut 45% this year, down to $12 million as a yearly budget. And this is the National Genomic Medicine Institute, having the same funding as a well-funded startup in the United States or Europe. So when people tell me, okay, why isn't the Mexican government tackling this issue? It really comes from I think a cultural, an infrastructural, and clearly a lack of vision into the potential that genomic medicine may bring in terms of improving patient care and drug discovery. So another option, and what most researchers in the region have resorted to, is parachute science, which is basically a paradigm that has been going on for centuries, in which foreign researchers come to underrepresented areas and essentially extract biological insights or gain be it through innovation or commercial gain in their home countries, right? So there's a picture of Howard Schultz, a renowned Harvard ethnobotanist. You can read all about his fascinating work in the Amazon. But what's most appalling is that a lot of his research and a lot of his discoveries were done on centuries of ethnobotanical knowledge that was passed down generation after generation by Amazonian tribes, to which received little to no benefit from these insights. In terms of genomic research, I think it's naive to say that this is not currently happening. Today, many genetic studies, particularly with indigenous populations, even within underrepresented countries, suffer from some degree of parachute science, where research participants are really considered as sample providers and data providers, rather than actual participants in the research. So something that we aspire to do in Omica as a community-driven biobank is to engage in what's called community-based participatory research. And community-based participatory research flips this paradigm on its head and really sees research participants as active participants in the research. This is mostly based on a protocol called the Nagoya Protocol, which was established by the UN a couple of decades ago to overcome the challenges around biopiracy and parachute science by creating fair and equitable benefit sharing agreements between researchers from the global north and communities, research participants in the global south. And this protocol has been mostly constrained or directed towards the exploitation of biological resources in the sense of plants or active compounds or seeds, etc., with the most successful use case being the rooibos tea in South Africa, I believe, in which the communities that have stewarded this biological resource are actually partners in the commercialization, the international commercialization of their genetic resource. And to us, our question is, how might we apply the learnings of the Nagoya Protocol towards human genomics, right? There's nothing more intimate than one's own genome. So why are we freely giving it away as participants and as researchers? How are we just freely asking people to entrust us with the ownership over their most personal data? So really what we're trying to see is reframing everything as participants, as researchers. And this involves tapping the community and allowing them to ask the questions that most matter to them. Not leading genomic studies from a top-down view of pharmaceutical-led R&D because there's a certain market or a certain disease area that is of commercial interest, but rather, what are the questions that are most important to these communities in terms of their health and their well-being? And most importantly, how to ask those questions. What is the adequate way of using their data in a way that is fair and equitable to all? And one of the key aspects of this new paradigm is redefining data custody versus data ownership. And this is something that as research organizations and as biobanks, we really need to ask ourselves and really innovate in the way that we perceive ownership or custody over samples and data, given that this data should be the sovereignty of each community and each individual that participates in research. So how might we reframe the ownership model and the economic incentives around genomic research in a way that positions us researchers as data custodians while giving participants data ownership. And to do that, Omica has really been focused on building a tech stack along with a number of partners. We see really genomics as being one of the best applications for blockchain technology, given its ability to deliver transparency and traceability over data usage. We see the main three things that a community-led biobank needs to have is increased transparency, for people to know where their data is and how it's being used. At the same time, providing evidence of provenance to allow researchers to know where their data comes from and whether their data was acquired ethically or not ethically, and what provisions are tied to that data. This requires a lot of innovation in terms of how does one harmonize consent agreements, right? So for instance, if Lygia and I are to work together, how do we make sure that the consents that we collect in Mexico are verbatim to the consents that she is collecting in Brazil, that allow for the same research provisions, but that allow for the same benefit sharing agreements with local communities. Benefit sharing has historically been very challenging, given the fact that it is very difficult to quantify the value of genomic data. A lot of times benefit sharing constitutes, giving patients a free blood sample or a free blood test or something like that. We believe that through a data ownership versus data custodianship model, we can engage participants to really be a part of a global data corpus. And this is the vision that we would like to suggest today, which is creating a global genomics data cooperative, where everybody owns their data, yet chooses to participate in research. And this is really the philosophy that we've built Omica on, which is that the whole is greater than the sum of its parts. And that the more transparency and the more traceability and the more generous we are with research participants, the greater data will be available, and the more we will advance as a whole. For that, we're currently partnering with a number of patient communities, local research groups, and of course, life science organizations. And we're just very excited to be here. We see Lifebit as a crucial piece in building this global genomics data cooperative. So just really excited to be here, and thank you so much.
Oh, that's brilliant. Okay, really insightful. So you talk a lot about benefit sharing in regards of the data ownership and the financial benefits that eventually arise from data to be funneled back to the actual owners of the data, which are of course the participants. Are there any other aspects of bringing back research, the benefits of research insights to local communities that you guys look at?
Yeah, I think there's tremendous biases, and not only biases in the data itself. When there's a lack of diversity, there's biases in the data itself. But we believe that there's also biases in the questions that are being asked, right? So if the entire life science, precision medicine research is guided through questions by people, sitting in London, the direction that precision medicine research is going to take is going to be biased to that one person's or that group's perspective, right? So we see that in orphan diseases, which in and of themselves are not orphan because they still affect thousands of people. They're simply considered orphan because the people who suffer from them don't have a voice. So I think the most interesting aspect of community-based participatory research is that it not only levels the playing field, but I think it will unlock tremendous amount of innovation opportunities and really valuable scientific progress.
Absolutely, I couldn't agree more. So thank you for now. We'll be getting back to you during the panel discussion. And as a brief reminder to the audience, you're very welcome to drop some questions into the Q&A box here in Zoom, so that we have a whole list of audience questions. We already had a whole flow of questions coming in, but please make sure to put yours in so we try to get to all of you. But for now, we are traveling back over the Atlantic here to our home in London and over to our good friends at Genomics England. And it's my pleasure to introduce Dr. Matt Silver, who has recently joined Genomics England as the lead genomic data scientist focusing on the Diverse Data Initiative. Matt holds honorary academic positions such as Senior Research Fellow in the Nutrition and Planetary Health theme at the Medical Research Council Unit, the Gambia, and as an Associate Professor in the Faculty of Epidemiology and Population Health at the London School of Hygiene and Tropical Medicine. And now over to you, Matt. Welcome on the webinar, and kindly share the screen from your end.
Thank you very much, Thorben and the rest of the team at Lifebit for giving me this opportunity to talk today. And I'm really going to echo many of the really excellent points that have already been made by Lygia and Victor, but from a probably slightly different genomic medicine perspective and from a UK perspective. So I should probably begin by explaining what Genomics England does for those who don't know. So Genomics England has a unique partnership with the National Health Service here in England to provide diagnostics through whole genome sequencing. And there's a particular focus at Genomics England on patients with rare disease and cancer. And this is explained, encapsulated by this first part of the loop you can see here, where we have the patient first reporting to the doctor with a condition, who can order a DNA sequence, the whole genome to be sequenced. This then goes through our bioinformatics pipelines here at Genomics England, and we can deliver hopefully useful information back to the doctor, and that gets reported back to the patient. But a key part of our mission also is to drive research in genomics. And we're able to do this because of the many patients and their families who opt to share their de-identified genomic and health data for research. And the idea is that over time, this healthcare data will generate new insights that can ultimately help to improve diagnoses and treatments for patients in the National Health Service. Now, within Genomics England, the Diverse Data Initiative has the aim of ensuring that these benefits, the benefits of genomic medicine, are shared equitably across the communities that make up England. And for those who are not familiar with the makeup of the UK population, we live in an ethnically diverse country with large numbers of people from Asian, African, Caribbean ancestries, and others, of course. And you can see here from this UK national statistics data that these numbers have grown substantially over the past 10 years here. So, of course, there are many reasons why health inequalities exist, and in particular why people from minority ethnic communities often suffer from poor health outcomes in the UK. And I think this is a nice schematic. So this schematic actually focuses on the application of AI to healthcare during the COVID-19 pandemic, but I think it captures sources of discrimination and bias, which we've already heard about from Victor, that apply just as much to genomic medicine and indeed healthcare more widely. So setting aside issues of wealth inequality and poverty, which we know are key drivers of poor health in the UK and of course in many, including in rich countries around the world, there's the issue of unequal access to healthcare, and bias is right at the start of the clinical pathway. And there are also issues of inequality in the use and application of medical knowledge in the form of treatments and access to cutting-edge technologies. But the major focus of our team's work within the Diverse Data Initiative is to tackle issues of bias in terms of the genomic data. That is the bedrock of discoveries, of course, in genomic medicine. And also to try to understand elements of bias that can creep into the systems that we use for genomic diagnosis, for example, when we don't take proper account of the genomic diversity that results from our varied ancestral histories. So you've already seen this plot, which you see in many of talks on diverse data in genomics from Lygia. And you know, most of us here know the extraordinary overrepresentation of people of European ancestry, in this case in participants in genome-wide association studies. And in fact, the number of genome-wide studies in non-European populations is actually increasing. You know, there's more diversity in studies, although there's still a dominance of European ancestries. But of course, when we look at the number of individuals who are participating in these studies, then this huge bias becomes clear. And we've already discussed this this evening, how the very small numbers of individuals amongst this group from major worldwide populations, including Africans, Latin Americans, and so on. And of course, also the admixed populations, which we heard in Brazil and Mexico, which we've already heard described, which are, you know, have the potential to give us fascinating insights into the genome and its impact on biology because of this these unique genomic backgrounds from admixed populations. So this lack of diversity has consequences in many fields of genomic medicine. And I'm going to go into a few examples here. So we can take polygenic risk scores as a first example. So polygenic risk scores predict complex traits like blood pressure or risk of type 2 diabetes from genetic data. And you can see from this plot looking at PRS predictions here for 17 different blood and anthropometric traits from the UK Biobank data. So if we take the prediction accuracy in Europeans as a baseline, we can see that these PRS perform rather poorly in individuals from different ancestries, most notably in Africans, where there's an average 4.5-fold lower prediction accuracy in this data. And once again, the problem here is a lack of training data from people from diverse ancestries. And this matters in a world where polygenic risk scores could be used to stratify patients into groups for preventative screening or treatment. So in this illustration, you can see that we can quite effectively target screening programs, for example, in breast cancer or prostate cancer, or we can target treatments here, statins in coronary heart disease, if we target individuals who fall into the highest polygenic risk strata. Now, the evidence that polygenic risk scores provide clinical utility over and above the more usual ways of measuring risk, family history or clinical biomarkers, is pretty limited at present. But I think this is quite likely to change in coming years. So this poor transferability of polygenic risk scores across populations starts to become a major concern. And the genomic data diversity problem extends to rare diseases too. So here's an example from cystic fibrosis, where there's a lot of allelic diversity amongst known cystic fibrosis causing variants in the key CFTR gene. And there are large numbers of actually ancestry-specific variants in this gene, which points to the need for screening panels to take account of these differences to avoid delayed diagnosis and increase morbidity in patients from diverse non-European ancestries. And here's one more example from the rapidly developing field of pharmacogenomics. So there are now numerous examples of genetic differences between populations that influence the way that we respond to drugs. And this example is of the CYP2D6 gene, where loss of function variance affects the body's ability to metabolize codeine, which is, of course, an important pain relief drug. And we can see here there's a wide variation in the ability to metabolize codeine, and this is due to differences in the global distribution of CYP2D6 variants, meaning that the optimal dose for pain relief also varies. And in certain ancestry groups, including one in Algeria, these include individuals who are codeine ultra metabolizers, and for these individuals, a standard dose of codeine can actually be seriously toxic. And this problem is exacerbated by the fact that we can see that the prevalence of non-normal metabolizers is actually unknown throughout much of the world. So I hope what all these examples show, and what we've already heard from the earlier talks, is that data are not neutral. And I think this is encapsulated very nicely from in this quote from the American sociologist Ruha Benjamin. Data and technologies are socially constructed and embed design decisions, assumptions, values, and ideologies that can be discriminatory and generate social harm. And as we've seen, this extends to inequalities in health outcomes related to genomic medicine. So what are we doing to address some of these issues in the Diverse Data Initiative? Well, a key priority for us is to increase the amount of genomic data from UK underrepresented groups and in neglected diseases in the UK, and make this data available to researchers to ensure that everyone can benefit from genomic research. But as Victor has already highlighted, central to all of this is the absolute necessity of earning the trust of the individuals who would like to participate and contribute their genomic and health data. And an example of this within Genomics England is we've got a major program that we're driving in partnership with others on sickle cell disease. So the aim here is to drive improvements in health outcomes by establishing a large genomic data set to research the genetic factors influencing the severity and progression of sickle cell. But importantly, we need to recognize that many people with this disease in the UK may have understandable reasons to distrust the medical establishment and medical research in general. So we need to work hard to build that trust for the initiative to be a success. And one way we're doing this is through engaging with patients to understand their priorities and concerns and make sure that these are heard and taken on board by the research community. Another aspect of our work is analysing as an analysis to understand the potential influence on ancestry on the existing whole genome sequencing diagnostic pipelines that we run here in Genomics England and in other areas such as pharmacogenomics. So to finish, I'll just give an example of this from some preliminary analysis we've done using data from Genomics England's 100,000 Genomes project. So this project aimed to uncover new diagnoses and improve treatments for patients with rare disease and cancer. And this is an ongoing endeavour. The project has already returned diagnoses and clinically actionable results for both rare disease and cancer patients. And in the rare disease diagnostic pipeline, so this works by identifying genetic variants ranked in order of priority from a clinical perspective, ranks from tier 1 to tier 3. And what this plot shows is that if we group these 100,000 genomes rare disease participants into genetically inferred ancestry groups, then individuals with non-European ancestry, so Africans here, South Asians and East Asians, have significantly more tiered variants reported by the rare disease variant calling pipeline. And a preliminary analysis by Sam Tauman in our group suggests that many of these additional reported variants could be relatively common in certain ancestry groups. So this means that they should have been filtered out, as they are most likely false positives that are very unlikely to be driving the severe early onset conditions that we find amongst 100,000 genome rare disease patients. And this increased number of false positive variants could in turn influence a clinician's ability to identify the true causal variants for a disease. So it's introducing biases in the pipeline related to ancestry potentially. And once again, the underlying issue here is that allele frequencies from discrete subpopulations in Africa and elsewhere are largely missing from public data sets such as NOMAD, and therefore haven't yet been incorporated into our pipelines. And this is supported by further analysis that Sam has done, where he's been able to look at frequencies in rare UK ancestry subgroups present in our data. And he found that many of these variants are present at relatively high frequencies in East Africans, for example. So finally, a similar analysis has been conducted on the cancer variant prioritization pipeline by TWI in our group. So Twee looked at germline variants identified in non-tumor tissue from cancer patients, and she found that the cancer pipeline also identified increased numbers of variants in non-Europeans that were putatively linked to cancer risk. So again, this could be linked to variants that are present at relatively high frequencies in certain populations that should in fact be filtered out. So again, leading to an increase in potentially false positive variants, as we saw in the rare disease case. So I hope what I've presented and what others have presented has persuaded you, if you didn't already need persuading, that genomics has a diversity problem, but that we and obviously many others around the world are working hard both to identify the gaps that need to be filled and find ways to tackle the resulting inequalities in terms of genomic healthcare and diagnostics. So I'll just thank the rest of my diverse data team and others in Genomics England who've supported and contributed to this work, and thank you very much for listening.
That was fantastic, and thank you so much, Matt. One burning question that I have, because we heard obviously a lot about in your presentation and previous ones about the inequality that it is that in existing genomic studies. The question is, what would you speculate are actually the main causes for that inequality?
Well, I think when you speak about inequality in genomic healthcare around the world, I think obviously the key issue is actually access to genomic medicine. So in most countries, you know, we're fortunate in the UK to have this kind of process for getting whole genome sequencing and diagnostics from that. But there's no access, of course, in most parts of the world. And I guess we can only hope that this will improve as the costs for rapid genetic testing, for example, come down. But I think there's also certainly even in a relatively wealthy country like the UK, there's also issues of engagement from certain communities and there are historic reasons why some communities might be suspicious of engaging with particular types of healthcare, and we saw that in the COVID pandemic with vaccine hesitancy, for example. And then, of course, the last big reason, I guess, is this problem of the missing data. So as Lygia said, we don't even understand how ancestry impacts the biology of disease across diverse ancestries. So there's just so much knowledge that's missing if we want to properly be able to diagnose diseases, understand prognosis and treatment. So that's the missing data gap again.
Absolutely. Thanks for diving into that a little bit, and we'll get back to you as part of the panel. Again, reminder for any of our viewers who would like to add to the question list, if you could pop them into the Q&A box in Zoom, not into the comments, in the Q&A box, please. But now we're moving to our very own Dr. Maria Dunford, who's the CEO and co-founder of Lifebit. Maria has been a lifelong biotech researcher, scientist, and entrepreneur who's actually founded several companies, including the Innovation Forum in Barcelona before she launched Lifebit and helped scale it into the business that it is right now. And with this, over to Maria.
Thank you, Torben. And thanks, everyone. Thanks, the incredible Lifebit team, for organizing this and for even inviting me onto this webinar. So I will just basically just repeat everything that was already echoed and very wonderfully explained by the three former speakers. We already effectively know that data can save lives, right? We've seen, especially the deep clinical and genomic data, speed up actually diagnosis by even up to 5 times. Actually, we are now able to actually diagnose rare disease patients and deliver accurate diagnosis. We've seen almost also 3 times improvements in cancer diagnosis, as well as even treatment. We've also seen this data help deliver more successful drugs. Actually, drugs that are based on genetic information are twice more likely to succeed on clinical trials. And also another striking number is that more than now 60% of FDA, so more than 2/3 of the drugs actually that are getting approvals from FDAs, are based on genetic information. So are drugs that are more precise, more tailored to different genetic backgrounds and biomarkers. And least but not last, or last but not least, definitely also this data helps us improve our understanding of diseases. Already studies that were done with the Genomics England COVID data, among other data, where pretty much the analysis that helped us identify 5 genetic markers that are influencing or were influencing the response to COVID, and we're explaining why some individual will be getting COVID and almost be asymptomatic, while others will be ending up in intensive care unit. So therefore, connecting this data is critical. It is also critical to connect this data because it has also been shown in various studies that when you actually increase this data by a 10-fold, you're actually increasing results and finding in terms of genetic associations by up to 100-fold. Unfortunately, the most diverse data are at the same time the most sensitive data. And it is more often than not that more than 95% of this data is completely inaccessible and very difficult to use, because this data is obviously distributed across the planet, and we actually seen that. It is therefore very hard to access and use. Because it is so sensitive, it is normally held on silos where actually restrict the access and the ability to, and limit the ability to run certain analysis over this data. Of course, we also heard it before that a lot of time this data comes in completely different formats and standards, therefore it's very difficult to combine. And as such, what we end up having is, in the end of the day, for any given disease that we are actually studying or breakdown of that disease, we end up not having enough patients and enough diversity for this data, basically, and for that given disease. So the solution to this is to try to analyze data in place. After, of course, we've actually standardized this data and we agreed on global standards, like, for example, the OMOP data standard, where we actually standardize this data. And there is only one type of technology that can allow us to actually analyze data in place, and that is federation. And federation is technology that allows for full usability of the data, while at the same time enforcing security by designs rather than security by certificates and agreements. And what we are now actually seeing, especially obviously that's what a big area where Lifebit is working with all of it, especially big pharma clients, is major pharmaceutical companies like Boehringer Ingelheim actually using federation to be able to connect their internal data alongside with the data of different other biobanks that are across the globe, and be able to actually get one virtual view of this data, which allows to run one type of query and then get the answer across all of these data. Like, for example, women over 60 with cancer and particular mutations on certain genes. And then the answer that comes back is that you can find the subset, you can say find 100 women, let's say within Genomics England or within GenT or within Omica, that actually can have fit that effectively phenotypic and genetic criteria that are being applied. But from there, the biggest thing is that now you can actually kickstart one analysis, and that analysis can be run in a distributed fashion where the data is, rather than trying to force the data outside of their secure environments, and effectively take the results from the different locations and then combine those results in order to give them back to the data consumers, which are, in this case, Boehringer Ingelheim and other pharma like Boehringer. And federation is not just driving more and better connectivity and usability of the data. It is also driving faster scientific and discoveries and greater insights. And also, as Victor mentioned before, it's also driving a new data economy, or better put, a precision is defining actually the economy of precision medicine, which in reality is a data economy. And it is helping the entire world to actually create for the first time one universal and more precise medical system that will enable that will democratize medicine for all globally. And with that, I would really like to thank you again.
Thank you very much, Maria. Fantastic. And if I can now ask our other virtual panelists to come back on, there's not an immediate need for you to do anything. If you just stay muted, we'll be going through a few questions or hopefully a lively discussion. And again, reminder to all the folks who are viewing, put any of your questions, please, in the Q&A box here in Zoom so that we can get to them at the end. And with that, the first question, actually, I'm throwing it straight back at you, Maria. What do you think is the biggest hurdle to overcome to enable more diverse data research? Can you just, address that?
So, as I said before, the technology is already here, and it is called federation. The faster we're able to actually adopt such technologies, and as also Victor said, we're able to create a global network of connected data, where we really separate, where every owner or custodian organization of this data comes in as equal into this big precision medicine and data alliance, right? I think that definitely will accelerate diverse data research. Obviously, the other thing I think, and that was also mentioned by Matt, we really need to do better job in order to inform and educate the different populations so that they can participate in such programs, right? Effectively, we need more data in order to have more connected data and more diverse data. So I think those are the two major things.
And more data, unfortunately, often is very expensive. And Victor, you mentioned how on a federal level, funding instead of being increased, it has been cut. Of course, that is the situation we're seeing. The moment you leave, maybe G7, maybe G20 countries, right? There isn't that kind of funding available that we are very blessed to see here in the UK. So maybe you can address a little bit that funding situation and how you're thinking about getting a program like Omica.bio and other related programs off the ground.
Yeah, of course. I mean, I think that's one of the foundational infrastructural differences between the ecosystem that we're seeing in our countries and the ecosystem that we're seeing in the very life science progressive countries, so to speak. And it's the fact that we are missing this key foundational element of public research, right? Where if you see the amount of innovation that has been built atop the UK Biobank, you see how that public initiative unlocked a tremendous amount of value for the global population, even, or projects like FinGen, or basically all of the public biobanks. When it comes to Latin America, I think we really depend on private-public partnerships in a way where we do need to develop very innovative economic models that can finance this long-term foundational research while also being wary of this parachute science biopiracy angle that I spoke about. So I think is a very fine line between finding commercial partners and aligning those incentives while at the same time safeguarding the community's integrity, sovereignty, and data, right? That the benefits are actually provided to the communities of origin, not only on the commercial side.
Absolutely. And interestingly, some of the cohorts here in the UK and Finland that you mentioned have actually been part funded by industry, right? By pharma companies. So they form a big part of this, of getting these programs off the ground. Obviously, it's to be done right. And Lygia, maybe to you to attract those global collaborations with industry, with pharma, with scientists at large. So how can these collaborations support drug discovery in your view, and how can the data specifically out of Brazil support that?
So, Victor, I relate completely to what you're saying in terms of not only, I mean, funding but having science as a priority and investment in science as a priority by our governments. We have a lot of things to organize in our countries, and sometimes different governments don't have the vision of the importance of science funding. And that's why I pivoted for the first time. I've been an academic my whole life, and I pivoted into trying to solve this with a different model, which is with the startup and relying now on investors and partnerships with pharma and biotechs, because it will be for everybody's interest to have these diverse data available. This will foster a lot of research within academia and outside of academia. And I think that at the end of the day, everybody will benefit from this. But as you said, there is this fine line where, are you selling Brazilian genomes to the big bad wolf of industry? And we really have to be careful with that, because we also want to bring development to Brazil as well. We want that our initiative, brings, it fosters innovation in Brazil, like the Genomics England did with England. And so we are now starting to talk to different companies and players about how can we generate the genomic data faster, because this is building the cohort strategically, it's very challenging doing all that. But in terms of generating the genomic data, the big challenge there is funding for that. So now we're discussing ways that we can partner with different groups to sponsor the sequencing of the samples we already have, so that everybody can start working with more diverse data.
Lygia, you kind of foresaw my question. You mentioned Genomics England already. Matt, Genomics England has long been a pioneer when it comes to collaborations with industry, with the pharma sector. Picking up on that fine line that Victor mentioned, how does Genomics England navigate that fine line and actually enable research also from the pharmaceutical sector over the data that GEL safeguards?
Well, Thorben, if you don't mind, I think I might pass on that question, because I'm rather new here and I've been very little involved with that side of our work. But if you don't mind, what I thought I might do is contribute my previous experience working in Africa, in the Gambia, because I think it's very pertinent to the discussion we're having. I think a key thing for really low income countries like the Gambia is a very poor country. What we really need to do is build capacity and promote learning and so that we can have indigenous scientists and genomics experts, bioinformaticians from the countries themselves, because that's the only way that the resources are going to be, we're going to build trust with the communities, but also the political environment can change, and that they can attract the resources, attract the investment to build their own indigenous science sector. I think that's the key to genomic, this big missing gap in terms of African data to be filled, is that we need to build the capacity there. And in my former institution, we were working very hard on that, funding bachelor's degrees, master's degrees, and a lot of PhDs too. But the problem we had was that many of the trainees, the PhD students we have, then leave and go and work in wealthy countries. So it really is a huge challenge, but I think capacity building is key.
Absolutely. That's something we see time and time again, that the projects that we work with globally want to see a local ecosystem, local knowledge, knowledge transfer to the local communities, so that gets in-house into the country, if you will. And I think that makes it more sustainable. and also helps kind of selling these programs politically. And on that note, Victor, you talked about the need for transparency to gain the trust in the community, in the population. So how do you go, and how do you suggest both Mexico as well as other places should go about creating that greater transparency around diverse data research, especially when it's a pretty complex field, right? Yeah.
Absolutely. I think we really need to support ourselves with technology. At the end of the day, there's amazing technologies being developed every single week. It appears that there's bounds and leaps in our technological, namely computational capabilities. And I think, at least to me, what's most interesting about the federated data model is that it lends itself to add a transparency layer where people know where their data lives. And I think that even to me, if I'm a participant in a research study, if I'm entrusting somebody with this incredibly personal data, I want to know where it is, and I want to know what it's being used for. And even if you can give optionality, so this, I think one of the core issues that we have as researchers is these static consent agreements. And I think the evolution towards dynamic consent through technological platforms that enable people to be consistently engaged can unlock a lot of possibilities in terms of not only giving people control over their data, transparency and choice, but also creating a means to continuously engage with participants for additional research. Right. So let's say that your original consent only covered genomic sequencing, but in 3 years, I don't know, single cell is going to become so cheap that you want to do it on your existing samples. There has to be a way to circumvent the logistical need to rebuild cohorts and re-recruit patients, and really use biorepositories as a dynamic source of future-proof data. And I think that's going to come with really adding layers to that data federation tech stack that enables people to be much more engaged in a much more dynamic way.
Absolutely fantastic. And on the point of federation, Lygia, you've been navigating the field of collaborative platforms across the world. So, you've been in this space for a very long time. Do you see governments around the world engaging really with the benefits of federation? Do you see that that's moving?
I see that is moving, and Maria's numbers are very compelling to show the benefit of doing that, of the synergy that is created when you add different data. And it's all about really you having an environment where that can be shared in a very safe and controlled way. So if we have the technology for that, I mean, this must happen.
Well, I only agree, of course. And as one last question to the panel before we move to the audience Q&A, maybe to Maria, what do you think personally is the most exciting opportunity, whether it's in research or in technology, that will help with the challenge of the lack of diversity in genomics in the future?
I mean, there are a lot of exciting opportunities, but and I think so. I think definitely looking, especially how we can attract different types of populations, right, for different types of research, and we're actually seeing some of these things emerging, right. We know that there are, as we're saying, certain rare diseases, orphan diseases, or diseases that are more specific to certain genetic backgrounds. I think actually using that as a really great excuse to apply and enable national and international funding. I think that is what we should try to educate, right? Because let's face it, there is disparity in the funds that we actually see across the world. But if we can at least raise awareness that for certain diseases, we need data and we need them yesterday, and then try to drive a campaign, if you like, where we say like, okay, can we please fund these diseases, right? And the generation of data for these particular diseases. I think that will I will definitely solve part of the funding problem that we see across the world. And at the same time, I do believe that the world has become smarter, especially politicians after COVID, because now they understand that for certain diseases, truly data is the answer. And if you do not fund the research and the efforts needed to generate this data, then you're not going to go very far, right? I think that at least is something that I personally try to drive as an agenda. So hopefully it will help.
Absolutely. And now I'm excited to actually have a look at numerous audience questions. We might not be able to get to all of those, but thank you for those who provided them. I believe they're coming up on the screen just now. So the first question, and I believe that's a good question for Victor, is the question is more around what are the data sharing restrictions of that model when it comes to data or metadata linkage of other national data or data from similar studies at a global level? And thank you very much for that question. Maybe I thought it straight away to Victor. What do you think? And if anybody else wants to chime in, please go ahead.
Yeah, I think this is related to my last answer around really leveraging technology to see where the data lives and allowing people to choose what they want to participate in and what they don't want to participate in. And I think one of the biggest issues around regulating that was the fact that once genomic data leaves your server, it's out of your server, right? And what's going to stop that data from moving around and flying around? So hopefully these federated data networks enable a level of control over where data lives and how it's shared and how it's used that will ultimately kick back personal control over how your data is used. And that really forces, by giving people and participants control over that data, it really aligns incentives in a way that research is really community driven. And it's really geared towards questions that matter to people. So it's almost like an analog to like a crowd-funded genetic research, right? Where you pay with your data rather than with your dollars. But yeah, I think that's something that is within our grasp, but will definitely require a lot of tooling.
And if I can ask further for maybe both Matt at GEL, Lygia, but also Maria, like to make data from these diverse cohorts comparable, what is a solution for that? So how do you compare very different sets of data? Who wants to have a go at this?
Well, if I may, I'll come in there. So one issue is the different platforms. So we know that for most GWAS studies, for example, the economical solution has been for more than 10 years or decades now is to use genotyping arrays. But at Genomics England, we are lucky to be able to have whole genome sequence data. And then, of course, there are emerging technologies, which I think are going to be increasingly important. For example, long read sequencing technologies from Nanopore and from PacBio, which are going to be vital for us to be able to target regions of the genome which we can't see now using short read sequencing and genotyping arrays. But of course, that then presents a problem of comparing across different kinds of data. So we'll have to get smart at doing that and to be able to combine different kinds of data. But we also need to get better at mapping genomes from diverse ancestries. We've used the same kind of reference for a very long time. So there are innovations happening there in terms of the pangenome, which I think promise to reveal a lot more of the missing diversity that we can't see at the moment. But all of this will involve sharing of data, and federated models, like we've been discussing, is one great way to do that. So these are challenges, but there are so many smart people around who are working on this now that we're developing the solutions.
Very good. And I think there's another question coming up on the screen. Let's see, that's from Joe. Whilst the bias of genomic data from an African context, the lack of expertise and experience in real-world data studies as opposed to clinical trials is a serious area of concern. And how can we circumvent that? Lygia, do you want to have a stab at that?
Well, I'll try. And what I was thinking while Matt was talking, that on top of all the challenges that he mentioned in terms of the genomic data, health data, capturing good health data in diverse countries is a challenge. I mean, what we're doing when we go to the electronic health records and trying to capture the health history of participants, that is really challenging. And I think this will be a price we will have to pay if we want to look at diversity. These are limitations that we have. So, you know, in countries not as well structured in terms of digital health, this is an added challenge to the effort.
Does anybody want to add in on this, challenges around that when combining data from these different sources? It's quite related. Maria, anything from your side?
So again, in order to actually bring this data together, data standardization is the most critical thing, right? We just need to standardize this data. It was also said before, the way, of course, so there are, I would say, in terms of to think about it, there are maybe 3 different problems, right? First of all, it's kind of like the collection of the data, right? Are we actually collecting the data in the same, let's say, protocols or ways, right? And when I say collecting data, logging them into our, obviously, because a lot of times how data is being logged into, let's say, a hospital or the healthcare system in UK, it might be different from how it is done in other countries, right? So already that can skew a lot of the data, right? Then how we generate the genomics data also plays a role, the protocol of the sequencing and everything we actually do around it. And then the last bit is the standardization, right? Once we get all of these genomic and clinical data, how do we actually bring them into one format that we can actually apply one unified query across all of the data and get an answer. And obviously, so those are 3 different problems which can be solved technically, or at least account for the different, let's say, biases or errors that we actually see within the data. But definitely those are a necessity, because if you don't kind of like pay attention in all these 3 areas, then it's very difficult to actually combine the data.
That is spot on, and I'm trying to see if you have time for one more question. I know we're coming up on the hour, and well, there's a straight answer question to you, Maria, again, is if Maria, can an interconnected federated model for human genomic data analysis include data provenance and traceability, and hence allow for direct benefit sharing back to participants? We heard a lot about this from Victor. Directly at you, Maria.
Yeah, 100%. I think Victor mentioned a lot of this, and I think he can also discuss more on that because I think he's more of even an expert of like doing blockchain and putting. So I think Victor said it best. Federation is just one layer. And then you can build other layers onto that federated technology that will further reinforce pretty much what you actually want to be doing, from cost recuperation to further traceability to a different kind of like attribution, right? And actually that can even become, I mean, at Lifebit, one of the, let's say, more R&D projects that we are working on is even to be able to measure the contribution of a particular data set to a machine learning model. Therefore, when a prediction becomes, then you can even almost attribute back kind of like basically what percentage of that success was attributed to the data was used on that federated machine learning model. And that doesn't necessarily have to do much with federation. Federation obviously is just the underlying layer allowing you to run that. But obviously, you're building other things on top. And that's just one example. And there are many other examples, many other things, right? So 100%, I think also was mentioned here before, but it's worth mentioning it again. And also maybe this is the biggest opportunity for us to actually advance connected data, is to educate the populations that that you're not commercializing or selling or giving away data. What you are commercializing is the ability to safely access and analyze this data in a way that there's never going to be an leakage or like a loss or a giveaway of individual level data, right? And I think then if you can do that, then it's easy then to convince people that if we're able to do that, then we're also able to attribute back how much your data is contributing to different analyses and to different potentially commercial success of different partners that are paying for access to that data, and therefore you can potentially attribute that back, right? But I'll stop here and maybe Victor can discuss more about it.
So unfortunately, while we could certainly end up spending the whole evening talking about this, it is by now evening in London, I will have to cut off at this point and mostly thank you, our wonderful speakers today, as well as our audience. So big thank you to Lygia, to Victor, to Matt and Maria. And I hope that if you in the audience have enjoyed today, please do scan this QR code on the screen to find out more about the topic, to find out more about our work that we're doing at Lifebit. And of course, we continue with these webinars, and our next webinar will be in September. So please do watch this space and dial back in and bring your friends. And of course, us and the team, we are always looking to improve. So do share your feedback also via the form that will be provided or has been provided to you already. And we have also recorded today's webinar. So for those who couldn't make it, those colleagues of yours, those friends, please do share the recording when it is up on our website from next week. But for now, a warm thank you and a thank you from London. Goodbye.